Hello, welcome to another practical class in bioinformatics. In this video, you will learn how to compute the reverse complement of a DNA sequence using Python. This is a very important concept in genetics and bioinformatics. So what is a reverse complement? DNA has 4 bases: adenine, thymine, cytosine, and guanine. A for adenine, T for thymine, C for cytosine, G for guanine. So reverse complement means replacing each base with its complement. For example, A pairs with T, C always pairs with G. So the reverse complement of A will be T. The reverse complement of C will be G. So in a DNA sequence where you have AT as the DNA sequence, the reverse complement of that DNA sequence will be now TA. In a situation where you have CG as a DNA sequence, the reverse complement of that DNA sequence will be now GC. So reverse complement means replacing each base with its pair. And someone may ask, why is it important? Why is reverse complement important? Reverse complement is important because it is used to compare DNA strands. It is also used in PCR primer design. Very important in primer design. And it also helps to identify genes on opposite strands. Then it is commonly used in sequence alignment and genome analysis. So these are the uses of reverse complement of a DNA sequence. Now let's move to the practical session. Just like we did for GC content, this is my desktop. Go to the search button, type Python. Python is already there. Click on it. This is the Python environment. This is where we are going to be writing our codes, just like we did for GC content. If you have not watched that video on calculation of GC content, you can go and watch it. The video is in my playlist. So now we are going to write a Python program that reverse a DNA sequence. I've written my code already. I'm not going to be writing it line by line because of time factor. So I've written my code already. So I will paste the code, then interpret it. So, okay. So this is my DNA sequence which I've pasted. I first of all define a DNA sequence. So we say DNA is equals to ATGC CGTA. That is our DNA sequence. It could be any sequence of your choice, but this is just an example. So after defining the DNA sequence, we also give what we call the complement rule. So I have to give a complement rule, and in that place I say complement is equals to, I use the curly bracket, complement is equals to A is the complement of T, then T is a complement of A, C is a complement of G, G is a complement of C. That is the rule I gave to the Python. Then to generate a complement, and I say complement sequence is equals to join complement. So I'm telling Python to join the complements together. Then after joining the complements, I'm telling Python to reverse it. So that is where I have reverse complement. Print result. So you can see our final result at the bottom. TACGGCAT. That is the complement of the DNA sequence that we imputed. So this is how to compute a reverse complement of a DNA sequence using Python. In our next video, we shall be dealing with DNA to RNA conversion using Python. If you find this video helpful, please subscribe and hit the like button. If you have questions, please drop it in the comment section.